Hi, I'm Kyvan Savage. I'm doing Biology A-Level, and in this video we are going to be looking at something that might come up in exam because it has not come up before. I also have not done a video on this, so I'm not going to skim over it. I'm going to do it in detail because I've been skimming over a couple of the few ones. Fingers crossed that this video uploads and doesn't... Oh, God, no one is sure over me. Hopefully this video uploads and doesn't not upload like loads of them have. Anyway, in this video we're going to evaluate the evidence for chemiosmosis. Now, what is chemiosmosis? Chemiosmosis is something that I hate. I think it is really bad. I can never remember what it's in. I never remember what the name of it is. If someone described it to me, I might actually be like, chemiosmosis is it maybe? And they'd be like, yeah, you know that. But if someone's like me describe what chemiosmosis is, I'm like, is it something to do with nerves? Yeah? Something to do with the animal kingdom? Oh, wait, no, that was last year. Oh, I don't know. And then when they tell me, I'm like, oh, yeah, I know that. So, I need to go through this. So, chemiosmosis, this is the key definition. I'm not even going to try and tell you this out of my brain because it will go wrong. So I'm going to read it from the book. Chemiosmosis is the diffusion of ions through a partially permeable membrane. It relates specifically to the flow of hydrogen ions across a membrane, which is coupled to the generation of ATP during respiration. In eukaryotic cells, the membrane is the inner mitochondrial membrane, and in prokaryotes, it's the self-surface membrane, which may increase surface area. Oh, it's so confusing. Okay, so what exactly happens in chemiosmosis? Well, as electrons flow along the electron transport chain, energy is released and used by coenzymes associated with some of the electron carriers to pump the protons across the intermembrane space. Okay, it's good I know that. This builds up the proton gradient, which is also a pH gradient, and an electrochemical gradient. Good. Thus, potential energy builds up in the intermembrane space. Okay, good. The hydrogen ions cannot diffuse through the lipid part of the inner membrane, but can diffuse through ion channels in it. These channels are associated with the enzyme ATP synthase, and the flow of hydrogen ions is chemiosmosis. Chemiosmosis is a flow of hydrogen ions. So there have been lots of experimental evidence and studies into this, and the first one was in the 1940s, and because by the 1940s, the link between the oxidation of sugars and the formation of ATP had made. And by the end of that decade, scientists knew that reduced NAD linked to metabolic pathways, such as the Krebs cycle, with the production of ATP. However, they didn't know that the biochemical mechanism by which ATP was made, and they thought that the energy associated with reduced NAD was first stored in high-energy intermediate chemicals before being made to use ATP. Investigations did not find such a high-energy intermediate. But then, during the 1960s, research teams were extracting mitochondria from cells and examining them. They used electron microscopes and special staining techniques. They could identify an outer and inner membrane with space between them, and they could see that the inner membrane was folded into cristae covered on the inner surface with many small mushroom-shaped particles. So then, we've got Mr. Peter Mitchell. By 1961, Peter Mitchell realised that the build-up of hydrogen ions on one side of a membrane would be a source of potential energy, and that the movement of ions across the membrane down an electrochemical gradient could provide the energy needed to power the formation of ATP from ADP and PI. He called this the chemiosmosis theory. The inner mitochondrial membrane is therefore an energy-transducing membrane. He postulated that the energy released from the transfer of electrons across the electron transport chain was used to pump hydrogen ions from the matrix to the intermembrane space, and that these protons then flowed through protein channels attached to enzymes. The kinetic energy, or the force of this flow, the proton motive force, drove the formation of ATP. Now, at first this theory was greeted with great scepticism, as it was really different from the idea of a high-energy intermediate compound. However, by 1978 there was loads of evidence supporting the theory, and Mitchell was actually awarded the Nobel Prize for Chemistry. Since then, scientists have established that stalked particles are ATP synthase enzymes, and have discovered how they function. It's also now known that some of the complexes in the electron transport chain have coenzymes, and these can use the energy released from electron transport to pump hydrogen ions across the membrane into the intermembrane space, where a proton or electrochemical gradient builds up. Now, there have also been some other studies, and there are evidence from these too. Some researchers treated isolated mitochondria by placing them in solutions of very low water potential, so that the outer membrane ruptured, releasing the contents of the intermembrane space. Then, by further treating the resulting mitoblasts, which are mitochondria without the outer membrane, with strong detergent, they could rupture the inner membrane and release the contents of the matrix. And this allowed them to identify where various enzymes in the mitochondria were, and to work out that the link reaction and the Krebs cycle take place in the matrix, while the electron transfer chain enzymes are embedded in the intermitochondrial membrane. Now, electron transfer in mitoblasts didn't produce any ATP, so they concluded that the intermembrane space was also involved. ATP wasn't made if the mushroom-shaped particles were removed from the inner membrane of the intact mitochondria. And ATP is not made in the presence of olymycin, which is an antibiotic. And now we know that that's because it's known to block the flow of protons through the ion channel part of the stalk particles. In intact mitochondria, the potential difference across the inner membrane was negative 200 MV. And that is more negative on the matrix side of the membrane than on the intermembrane space side of the membrane. The pH of the intermembrane space was also lower than of the matrix. There was also a large time lag, if you notice, between making a discovery and being awarded the prize. And during this time, other scientists repeated the work and carried out further research. And this meant that loads more evidence was gained to support Peter Mitchell's theory. The more studies are replicated with other scientists coming to the same conclusion, the more reliable it is. And by this time, the scientific community is able to accept a new theory, and therefore they judge how significant a discovery is. So that is super, super good. Now we have evaluated the evidence for chemiosmosis, and hope it doesn't come up in the exam because it's stupid. Anyway, yeah, good luck with your exam. I hope it goes really well.